Here we have part 6 of the immune system, page 773. And we're coming over here across the page and we're noting adaptive defenses. And of course, we're dealing with what I had just described to you as the humoral and cell-mediated immunity part of our program here in part 5. So now we're in part 6, and we're going to describe more details about that adaptive immunity that is specific, number 1. They're giving us some listings here to describe the active immunity. So it's going to recognize individual and particular pathogens. There's going to be small molecular characteristics of those pathogens that are just the right size for us to recognize, because they're just the right size for the antibody or immunoglobulin to bind to. It's systemic because we're dealing with the system of cardiovascular blood flow, and also the lymphatic system is implicated here. So the lymphatic system is going to be, how shall I say, crowded in its lymph nodes and other lymphoid tissues with lymphocytes of the type of the B cell persuasion, the T cell persuasion, and the macrophages. It's going to have memory. So in addition to producing the immunoglobulins of interest, there's going to be a subset of cells apart from the plasma cells, or actually the plasma cells, that will form a subset of memory. And in some cases, you need to have more memory cells, and you actually need to have a booster. Oftentimes, vaccines require more than one administration in order to have really, truly vivid active immunity. Okay, so with that in mind, we now focus our attention on to what is an antigen that might actually cause an immune response. So we're checking out over here an antigen. Now this could be a bacterial cell. It could be a viral particle. It could have numerous places on it. And do you notice that we have a situation here where one antigenic determinant has this shape, and so it's specific over here. Another antigenic determinant has this shape, yet another antigenic determinant has this shape. Right? How many antigenic determinants can we have? Here there's listed 3. We have the genetic potential of rearranging our genome in B cells and T cells to provide for 3 times 10 to the 8th different antigenic determinants. There's several stretches of DNA throughout our genetic code that can be rearranged at certain points. And so if you multiply together all of those different possible genetic differentiations, they come out to 3 times 10 to the 8th. So you can write out your zeros there and see how much that is. So that's the idea of what's going on here. Okay, so that's what our adaptability is. What is actually an antigen, and what's a hapten? I like to think of that as just happened to be there. Okay, so a true antigen is something that has certain properties. It has to do with size, and it has to do with how it's addressed by self, not self types of things. It has to appear on an antigen-presenting cell. It's the product of digestion of a pathogen or other materials. Okay, and so it has to have these 2 properties in order to be a complete or functional antigen. It has to have immunogenicity, which is to say that we're going to stimulate one of the arms or both of the arms of humoral and cytotoxic immunity, cell-mediated immunity and humoral immunity. Okay, so we're going to get antibodies from the deal, and we're going to get activated T cytotoxic cells from the deal. Also, it has to continue to be reactive, that is to say it can be found by those things that we've created. Did we create antibodies? Did we create a T cytotoxic cell? And then can that cell react? Can that antibody react or bind to? Some things are too small, some things too large. It has to actually fit the size of an immunoglobulin. Right? If it's too small, we describe it as a hapten. Something that actually, in another language that I'm not familiar with, this means grasp, but that is to say this is going to grasp a protein. This is an incomplete antigen until it attaches itself to a protein, and then it becomes a functional and complete antigen. And there are some common things in the environment that many of us do not respond to at all because they're just there, they're small, they're not meaningful. Okay, but they can, in some cases, in some individuals, bind to proteins and produce an antigen, a complete antigen, and that would include such things as penicillin. Small children actually have this problem more often than can be described, where penicillin binds to a protein and then becomes an active and systemic antigen. There's chemicals that are available in the environment that cause this to happen. There's poison ivy, which is an oleoresin of poison oak, poison ivy, and other plants of a similar variety. There's animal danders. So some people are allergic to cats. I suspect that there may be cats who are allergic to people, right? So the cat's looking at the other cat and saying, I like people. I just can't be in the same room with them. So that happens often. Anyway, so the idea that you can actually be allergic to hair danders, detergents sometimes can cause that to happen. There's cosmetics that eventually become, after long use, binding to proteins in the area of the eyes or the face, wherever the cosmetics are used. There's other common household items, products. I would like to refer you to food lectins, where some people are allergic to nut oils and so on. There could be some other foods that are less noticeable but more generally regarded as difficult. It's possible that there's, we already know that there's something called Clostridium difficile, which is an encapsulated fungal pathogen that lives in our gut, that when we have too much in the way of antibiotics, that wipes out the good flora that protects us. What's left oftentimes is the encapsulated Clostridium difficile, which then has its way with us once we stop taking the antibiotic, and it unfolds and creates all kinds of havoc. So the molecules that are present and available from that, the toxic molecules from C. difficile, can be a problem for people that develop colitis and so on. So let's take a look here at, they talk about antigenic determinants in some great other detail, but I think we've covered that topic adequately at this point, and we can press on. In some cases here, they're reiterating some things that we need to know, but one thing that's new and interesting in this particular page right here, 776, is where we address T-cell activation as a result of visiting the thymus. So visiting the thymus, which is an endocrine organ, it specializes in activation of T-cells between the age of birth and puberty, where it sort of finishes its job, and then by adulthood it goes away for the most part. Okay, so activation of T cells is required. Okay, then we have they talk here about positive and negative selection and self-tolerance. We need to have a major histocompatibility complex in order to have self-tolerance. That is to say, we need to be able to recognize our own name in the H and the major histocompatibility complex, often referred to as class 1 self-antigens. Class 2 self-antigens are unique to those, that constellation of cells that are going to be actually doing the immune system. That's sort of like the prime directive, that we don't mess with class 2 cells in any way whatsoever. So they're overly protected, whereas class 1 cells are pretty much all the other cells of our human body. This is an interesting photograph right here, a micrograph that shows a really vivid macrophage. Whenever you get to the place of having a dendritic cell that has this level of dendrites, this is extraordinarily active, particularly in the response to cancer.